Hi, I am Asipuraman Shah, and today we are going to talk about plant cell cultures. So, cell culture or plant tissue culture is the growth of plant cells, tissue, organs on artificially defined media under aseptic condition in a controlled environment. In this type, we can grow a whole plant from a single cell or its tissue. So, moving on, Uh, Tutipotency. It is a very important term to understand if we want to talk about tissue culture. So, potential or potency of a cell to develop into a complete organism or differentiate into any of its cells or tissues. Okay, tutipotency is the potency or potential of a cell or tissue that it de-differentiate from differentiated cells and then become a whole plant or its part. So we will discuss about differentiated cell and de-differentiated cell in later uh, in in other slides coming in coming slides. Pluripotent is derived from two Latin words. Tutus means entire, potent means potency. Many somatic plant cells, including some fully differentiated types, for example, leaf, mesophyll, have the capacity to regenerate into whole plants. This is an amazing developmental plasticity. You can say that tutipotency can relate with the plasticity, because if you know that plastic has the ability that it has elasticity. So plant cells or plants have that kind of elasticity that like they can be made from a single cell a whole plant. So it is just in plant that kind of totipotency that it can be grown from a single cell. Plant cells apart from most of their animal counterparts. In animal cells, it is very hard to make a whole organism or individual from a single cells, because animal cells or animals are not that much totipotent or plastic. Many plants have been regenerated from single cells, but all plant cells do not have this ability. Almost every plant can be regenerated from a single cell or its tissue, but it doesn't apply to each and every plant. Remember that plants at most stages of life cycle have some population of cells that are totipotent. Totipotency is of course also a property of a normal undifferentiated cell, for example in meristems, in animals stem cells. So going to make a whole individual plant from a single cell, first it has to be de-differentiated from differentiated form. The first step in expression of regenerative totipotency is for mature cells to re-enter the cell cycle and resume cell division, a process known as de-differentiation. As I told you earlier that first cell has to go to um, a de-differentiation, which is called uh, which can be explained or defined as cells to re-enter the cell cycle and resume cell division, a process known as de-differentiation. This may lead directly to organized development, where somatic embryos develop, direct embryogenesis, or formation of shoots or root, direct organogenesis takes place. Alternatively, maybe sorry there may be an intervening callus stage from which organized structures can later be introduced to develop, referred to as indirect organogenesis or indirect embryogenesis. Also, now we will just touch a bit of history that Heberland in 1982 first developed aseptic culture in a neutral solution. White in 1934 created first root culture. Gothret in 1939 first produced or made callus structure, callus culture. Nobcott in 1939 also formed the callus from carrot. Cooking in 1960. mm, uh, what did cooking in 1960 is Zymatic isolation of protoplast. It started protoplast and Morashik and School. It is very MS media, we call it as MS media, was first prepared by Morashik and School in 1962. Scooby Dooby Doo. Wessel and Helderbrand in 1983 regeneration from single cells. They do regeneration from single cells. So it was a small history. Aseptic techniques. So we need aseptic conditions there to produce a whole plant from a cell culture or tissue culture. So aseptic conditions are very important. Microorganisms like bacteria and fungi are very fast growing and can quickly overrun plant cells cultures and destroy them. You know that in media um, there is plenty of nutrients. So if any bacteria or fungi reach there or contaminate that media, they have the ability to reproduce quickly, and so they will run over the plant cells and culture and destroy them. So that's why we must ensure a very good aseptic condition, uh, complete aseptic condition. Therefore, to avoid contamination, tools, containers, media, explant, working environment, and growth room should be sterilized. Aseptic techniques, these are the list of aseptic techniques. We will talk about them step by step. Sterilization of culture rooms, transfer areas, UV light. We use UV light. Ethanol detergents. What we do in laminar flow hood, we have UV light UV light tube bulbs. So um, we on we switch on that bulbs, and we let that place because UV is very dangerous to us too. So every kind of bacteria or contaminants uh, damaged, or you can say that deteriorated by the UV light. So after that, yeah, ethanol or detergent. We wash our hand with ethanol before working uh, in the lab with cell culture or tissue cultures. Another sterilization of tools is very important. Culture vessels, like we have some tools, petri plates, or any things which is going to be used in the laminar flow hood or the experiment. So we need autoclave. In autoclave, you know that at 121 degrees centigrade, 1.05 kg per centimeter square for 20 minutes, in which the pressure and moisture, hot moisture, water, you can say that steam kills the every kind of contaminants. And dry heat can also be used 130 to 180 degrees Celsius for 2 to 4 hours. Sterilization of culture media is also important. It can be done in autoclave. Filter sterilization of sorry, unlike oxygen or cytokines, which are sensitive to the heat, so we can filter them for sterilization purpose. And sterilization of explant, we do, we take a plant, okay, the tissue or the cell which is taken from the parent parent plant is called explant, okay, because it is now no more the part of the plant, parent plant. So what we do, we take hydrogen peroxide or ethanol and these kind of disinfectants, and we wash them, we wash expand with them, so we get sterilized. So we will talk about you can see it, you can stop and see all these disinfectants and their use. Okay, so we will talk about the types of cell culture in our in next video. I will appreciate your comments and your suggestions. I will encourage you to comment, share, like, and please also subscribe to my channel. It will boost me to make more videos, and it will help me to make more videos for you. I hope you are understanding it.